threshold, we are decreasing the risk of toxicity. So what we've seen with that kind of molecules is that there is a high variability between the different individuals. So people have a lot of different clearances, volume, and everything. So the as variability implies a high variability of the exposure of each patient. And as we have a quite narrow therapeutic window, it means that for some of them, the flat dose is going to be good. For some of them, we're going to have a too high dose, which means that there is a high risk of toxicity. And if we have, for some of them, too high clearance, for example, there will be not enough exposure of it. So that's why therapeutic drug monitoring in that case makes a lot of sense. So the goal is not to change the current care, but to add some additional information to go toward that direction. So what do we mean with the tool that we have developed on some of these molecules? So more and more user-friendly usage of this information for clinical usage. So it's to help the decision and to guide the decision using information that we have at the hospital. In that case, what we have is that we have additional measurement of what is going on for the patient. So we have typically the drug at the last intake. And what we are able to do, we do that in several steps. We use pharmacometrics models with the pharmacometrics parameters to compute the actual PK profile. So the goal of this first step is to narrow the model not to the full population, but to the specific population that we are considering. So for example, if we have covariate, relevant covariates, like age, sex, weight, that are quite easy to take into account in a clinical care facility, and the individual measurements, we're able to narrow the variability of the information and to have a much better information on the PK parameters, not only being the parameters drawn from the population, but what we call the conditional distribution, meaning that it's this population subject to the measurement that we have. So based on that, we're able to really decrease the variability of the information. So what we have is the variability of the clearance and the volume, as we are not taking only the population into account, but adding covariates and measurements. The conditional distribution have a much lower variability. And with that information, we're able to simulate virtual patient as exactly the same one, where we are able to simulate new drug regimen corresponding to possible regimens in clinical care. So different doses, maybe sometime different interdose interval to be able to have a possible drug administration to the patient. And what we do with that information, we are simulating like we would do with a classical clinical trial. And based on that, we will be able to use this information to simulate this drug regimen and see what is the percentage of chance of being on that specific therapeutic range. So how does it work in practice? This is some screenshots of what we have in several hospitals in Europe and in the US. So in that case, it's a drug called sunitinib, which is administered for cancer. The clinician puts, or the pharmacometrician in the hospital put three kinds of administration information. The first one corresponds to the current care, so the current treatment. So to see, in that case, how many people will have this kind of information and what is the amount of treatment that will be provided. So this is the current clinical care. The second information that is applied is the last intake. So we're going to make a drug measurement, but we have to know when the drug was taken the last time. And in that case, we just define as a date and a time when it was received by the patient the last time. And finally, the drug measurement, again, a definition of time. So a time based on a date and a specific time in hour and minute, and the drug concentration. So in that case of the sunitinib, the concentration is both for the sunitinib and its metabolite. And with this information, what we are able to do, we have this point in black corresponding to the measured concentration. And the first thing that we are able to do is to determine a pharmacokinetic profile. So what is this current pharmacokinetic profile? It corresponds to the profile of the patient and its uncertainty and its variability due to the population and its uncertainty as we are reducing that from the pure population model to the conditional model. So based on that, we're able to draw the median and all the variability across the uncertainty that we have on this patient. So with this measurement, we're able to have this variability of profile. And what we see is that we have an estimated residual concentration corresponding to the sum of the parent and the metabolite that we estimate at 85 nanogram per milliliter. And what the studies in hospital care have shown is that the target range for concentration is between 50 and 100. Two things that are important in that part. First of all, it is a key element to see that we are not just taking the mean value or the most probable one, but provide also an information of the uncertainty and the distribution of its uncertainty. And in addition to that, what is typically done in hospitals is to take the measurement at the trough concentration, which can be very difficult from an operational point of view, to be sure that you have the specific date. And with this kind of solution, with pharmacometrics and simulations, we're able to remove this constraint and take more or less the drug measurement whenever we need. With this information, so we are able to have a lot of simulated individuals like the patient that we are looking at, and we are able to simulate new drug regimen corresponding to different doses. So in the case of the sunitinib, we have the reference treatment is 50 milligram per day, and we have the possibility to have a micro variation of 12.5 milligram, and we test the different dose regimen, and we are computing which one has the more probability to be in the target. So what we see in the presented case, which is very interesting, is that we see that we are with a high probability of being in the therapeutic range for this drug and this patient. However, we have the possibility to reduce the amount of treatment and increasing the chance of being in the therapeutic range. What it means by that is that even if we are already in the good therapeutic range, we can decrease the possibility of toxicity and also decrease the cost of the treatment. So finally, as a conclusion, we made that kind of evaluation on a lot of different patients and applied the therapeutic drug monitoring on 233 patients at a French hospital called the Hospital Cochin in Paris. And what we've seen is that if we are looking at what is the good dose suggestion based only in this information, only 16% of the patients we will keep exactly the classical drug recommendation in terms of, and we have 67% of the time we would need a lower drug amount, so typically 1 or 2 steps below, and 17% of the time we need additional dose to make sure the people are enough exposed to the drug. And what we've seen is that it's very convenient on a daily usage for the clinician, and that's a good help not to change the current care, but to use this additional information to make a better care for the patient at the end of the day. Thanks a lot. Any question?
Well, thank you very much, Jonathan. That was an excellent presentation. I don't think we have any questions from the audience at this point. I was just going to ask as a follow-up, I guess, you pointed out to us that 67% of the patients look to still require a dose adjustment, and it's a fairly significant amount. And I was just wondering, will you be looking further at perhaps covariates to try to better identify what some of the characteristics
Exactly.
are?
What we've seen in the studies with the hospital is that we have a lot of adaptation that is possible by seeing additional covariates like SAGE as age, weight, or classical covariate or creatinine clearance. And what we've seen also, especially in, I would say, real care, contrary to classical clinical trial, is that we have a very different population. Typically people who have more severe disease, people who are older, and also to follow them on a longer term than a classical 1 year or 2 year clinical trial. So it shows several things that we have the possibility to adapt because we have this possibility, and the typical care is a flat dose because it's much more simple to provide that as a classical care. But how do we use this information to make that more efficient? And how do we do that for not more complex but more concrete cohort corresponding to older people typically, or people who may have several covariates, much more dedicated at some values at some point, depending on the geographical area, to maybe older people and things like that. How can we add this information that has been most of the time treated during the clinical trial to make a better care for the patient in the hospitals?
Well, thank you. It's a really interesting project. So we're down to our last few seconds here. I just wanted to thank everyone for joining us for this session, but we'll close it out now, and Jonathan will be available at the panel discussion following this track at 2:15. Thank you.
Thank you very much.